All right. All right. Thank you, Monica, for the introduction. Good afternoon or good morning, everyone, depending on where you are. My name is Chou. I am an assistant professor here at Stanford. Last month, or I think November last year, me plus three of my colleagues, we are also on this event, and at that time we have a kind of a tag team in a way that to talk about what we are doing, and it's very honored that Monica invited me again to talk a little bit deeper about some projects what we are doing. So today I'm going to talk about one project we are doing that is basically with an aim to develop new insulin that can have potentially better clinical benefits and improve properties for people with type 1 diabetes.
Great.
Here's my disclosure. So I actually would like to acknowledge all the people here because it's been a while for us to for this project. So there are a lot of people involved, and these are people who are former people in the lab, some two former students and one former postdocs, and then these are the current people in our labs working on these projects. We have a lot of collaborators along the way. I will talk a little bit about that from more on the natural product side, and then to more structural biology, some in vivo testing, and then some biophysical testings here. And then here are our funding sources.
And it's really interesting, I would say, Danny, that, just to sort of highlight again that your background is organic chemistry. And so you're sort of one of the few in the field coming from that background, which makes you unique and I think uniquely positioned.
Yeah, good. Thank you. Yeah. So when we talk about the advance of insulin therapeutics, I really love this figure from one of the reviews from a few years ago in the way that it really comes a long way for basically all the people in the pharmaceutical industry, mostly from Novo, Lilly, and Sanofi, for them to develop a lot of different insulin analogs with different basic pharmacological properties, or I mean, we should say pharmacokinetic properties, from insulins that can be very long acting and all the way to insulins that can be very fast acting. And by controlling what's in the vial, the way they behave when they got injected in the subcutaneous depot area, in a way that some will be multihexameric, some will be more toward monomeric and dimeric in a way that they can be absorbed by the body faster, in the way that they created a lot of different combinations of different molecules that can be further utilized by people with diabetes to further improve the clinical outcome. And I also really like this review from a couple of years ago in the way that the authors are highlighting, what are the past challenges we have already solved, what's the current challenge people try to solve, and what's basically the future challenges of insulin that we hope what we can go for. So I mentioned to you that with all the developments of different insulin analogs, there are a lot of past challenges that people can now do a pretty good job in a way that try to make things act longer in the body or act faster. But still, there are some current challenges people want to go for. So, for example, people still want to go for something that's even longer acting, and we now know that there's insulin icodec from Novo that's currently in phase 3 trials, and then hopefully we can potentially see a weekly insulin available soon. There are, of course, a lot of efforts trying to go for insulins that can be very good for the closed loop systems, and then potentially even faster acting analogs there, along with some other features that people are trying to solve. And then there are also kind of more future things that probably still right now there's only a little bit in the clinical studies, and still need some time to reach to reach the community of people with diabetes. So, for example, glucose sensitive insulin is a one big area in the way that people are still trying to develop it. And then some of you may know that there is a preprint study, that means that probably still a paper that's under review from a team of scientists in Novo Nordisk. They have some exciting results trying to push for that. And there's also areas like liver-preferential insulin, because we know that for insulin to be secreted from pancreas, all of them will go to portal vein. So liver got the first pass, and they got basically the most exposure of insulin. But when we are doing subcutaneous injection, it's more systematic distribution, so it's pretty difficult to restore the high liver insulin exposure. So one of the key challenges to tackle in the future will be trying to develop something that's more liver preferential, along with lots of different things that we as a field will work together to tackle it. Today, what I'm going
Danny,
to
can
talk,
you unpack that a little
please.
bit when you say liver preferred insulins?
Yeah, so like what I say, one of the more important kind of rationale behind it is that when insulin got secreted from pancreas, everything will go to portal vein. So basically, everything will get delivered to liver before it reach out to the rest of the body through the systematic circulation. So basically, there's a fundamental difference between subcutaneous injection, which is basically, you will put it somewhere in your body in a way that everything will go through systematic circulation right after the absorption, versus in insulin secretion. I would say one of the benefits of say islet transplantation is that those islets can be put somewhere in the portal vein area in a way that when you are going for islet transplantation, you can potentially restore the liver insulin exposure a little bit more. But this is pretty difficult to do if you want to just have a subcutaneous injection, but still somewhat restore the liver, higher liver insulin exposure. And this will require potentially different approaches to go for that. One of the approaches I know that there's a company trying to do that is they try to basically pack the insulin into some sort of polymeric system, such that because of the size, more will go to liver, and then there's a way to achieve it. And then potentially, as technology advances, hopefully there will be more and more approaches. People can basically recapitulate the higher liver insulin exposure by just doing subcutaneous.
What company is that, if you don't mind me?
I believe it's called Diasong. If there's anyone in the crowd, they know a little bit more about this company, they can let me know. But I believe it's a company called Diasong, and then they are trying to develop something like this.
Interesting. I just wanted to kind of just flesh that out. Thank you.
Yes. So today I'm actually going to talk about our effort in developing faster insulin analogs, and then this is actually a pretty active field for people to think about it. And then I actually want to talk about this review from Dr. Hasey. I actually saw him in the attendance list here. So I really like this review. Like this clinical study published, because it provides a direct comparison between at that time they call it ultra rapid Lispro, and then or with the brand name Linjaf, and also Fiasp, which is basically the faster acting insulin aspart. It's a direct comparison between the two kind of faster acting insulin formulations versus their parent molecules, which is Humalog and the Novolog. So basically what I do is, in the two faster acting formulations, they add excipients in a way that to make them to absorb faster. And as you can see here, pretty clearly from the serum insulin level here. So we can see that indeed both FIASP and NINJAF, they can be absorbed much faster, especially in the first 30 minutes. You can see a pretty significant increase of the insulin absorption inside the blood vessel to go with it. So, of course, this is a very clear evidence to show that they are indeed faster acting. What is potentially even, or at least equally interesting, is that in terms of the duration of action and then basically the tail end, they actually look very similar in the way that basically the duration of action basically is the same time between the parent insulin analog and the faster insulin formulation there. And then this is potentially, and that's also something we really try to address in a way that this is one of the things we believe that still we need the field to address. And the reason why we thought if one can develop something that's with shorter duration of action that could be beneficial is because the post-meal hypoglycemia is still one of the main challenge for potentially those automatic insulin dosing system to address, because as you probably can see, if you inject the insulin in somewhere around, in the T equals to 0 range, even after 2 to 3 hours, you still see roughly a third to half of the insulin level left in the serum, and then potentially can still have a lot of glucose lowering effects in the body, and that's already 2 to 3 hours after the original injection. So this tail end effects is actually pretty difficult, for the algorithm to control, and then potentially to make the closed loop system to be even more aggressive to basically control the blood glucose levels in the tiny range. And this is really something we believe that if we can basically make something still go do subcutaneous injection, but with a much shorter duration of action, that could be something very beneficial. And that's kind of the direction we are thinking about it. There are lots of different potential explanation or rationale to explain why one is still seeing, even though they are faster acting for these formulations, but you are still seeing a similar duration of action between Humalog and NINJAF versus Novolog and FIASP. We believe the reason is because the insulin is still basically formulated as an hexamer, and then although we believe that once the Lispro insulin or the aspart insulin injected inside the body, they can be somewhere in the mix of dimer and monomer, and that's probably still a little bit difficult for them to kind of get out of the subcutaneous depot, and that potentially lead to the longer duration of action. This is a review written by my colleague here at Stanford, Eric Appel's group. They talk about potentially, when insulin formulated as an hexamer, but they actually develop some excipients in the way that they can potentially stabilize the monomeric form of insulin, and they are able to see something that's potentially much faster acting and also with much shorter duration of action. And this is basically the reason why we also believe that if we can actually develop something that's truly monomeric, and then this may be something that can lead to something with a faster onset action, but also shorter duration. And now to go back to think about the monomeric insulin feature, and then why we believe that could be helpful is because really in the subcutaneous depot area, once you inject the insulin in, it becomes a question of the biophysical properties of insulin multimerization. So basically, native insulin would still very easily form an hexameric form in the subcutaneous depot because the concentration is still quite high. But in the case of Humalog or Novolog, because of the dimer association state between the insulin is now not as strong. So basically, you can make it to more like a dimer or potentially more monomer, and then that's the reason why you will see more GAP absorbed in. And also, we are seeing a shorter duration of action between Humalog and Humulin here. What we are trying to demonstrate is potentially we can have a novel insulin in the way that they are always monomeric to begin with, and still with full bioactivity. We can basically go with something that can be much more faster to be absorbed and potentially also lead to a shorter duration of action. So basically our goal to begin with, I mean, this actually started 7-8 years ago, is can we actually develop monomeric insulin such that we can achieve both fast acting property and also a shorter duration, that means a faster offset action as well. So basically, this is basically the same concept, but I kind of make it with a easier schematic presentation here. And then the real idea is, can we actually make something that wouldn't, that will not become monomeric? So one of the known region that is highly involved in terms of the dimer formation region is this part in the C terminal of the insulin B chain. So there's a pretty strong anti-parallel beta strength interaction here. And then it's actually well known that if you can cleave this off, this will basically generate a insulin people call it disaccharide peptide. Basically, you cut the B23 to B30 off. You cut A amino acid off, or in short, it's called DOI. This is the insulin that's monomeric. Okay, but also long ago, people already know that if you cut this part off, you actually will lose potency by quite a lot. So basically, you will basically make the insulin to be not potent enough to be used as a drug for people with diabetes. And from a lot of structural biology work, now that we know that this region, in addition to forming insulin dimer and hexamer, they are also very important because they interact with the receptor. So such that when you delete this part off, their ability to engage with the receptor binding is now much weaker, and then that's why we are seeing a significant negative order change of the potency there. So this kind of make this thing that's quite difficult. And really, the reason why we started work on this is actually long ago, we actually happened to get involved with a molecule that's basically, it's a venom molecule that's secreted from these fish hunting conch snails. So basically, these are fish hunting conch snails in a way that they will secrete venom such that fish will stay there, and then fish won't move, and then as a way for them to do prey hunting. So most of the time, the molecule they found from the venom, they are either neurotoxins or ion channel blockers. That makes sense because you want to intoxicate the fish for them not to move. But this time, so two of my former colleagues at Utah, Professor Spivey and Professor Oliveira, they actually found a molecule looks like this. And then if you know insulin for a long time, you will know that they actually look quite similar to native human insulin sequences because they actually share some sequence homology in the A chain, not too much on the B chain, but also they have the same disulfide skeleton backbones. What's even more interesting is the venom insulin actually does not have the dimer formation region. So at that time, we make a prediction that this will be a monomeric insulin to begin with, and that's something quite interesting for us to study. So to begin with, in their seminal study, they actually make a biselenium analog of the venom insulin, and they were able to see that because they were trying to prove that it's a insulin that target fish in a way for fish not to move. So they can see that these venom insulin can reduce glucose levels in zebra fish. And you can also see that compared to the control, the one with the insulin there, they move much slower compared to the control in this way for them to kind of demonstrate the potential prey hunting behavior here. But the next question is, are they really monomeric? The answer is yes. So their association state is 1.05. That's pretty much means that they are monomeric to begin with. So it makes a lot of sense because it doesn't have the same dimer formation region. So they are indeed monomeric, and then this is actually quite different compared to a lot of different mammalian insulin sequences. Very early on, we actually found out that the Venom insulin actually doesn't have a very strong potency to a human insulin receptor, either binding or signaling. So somewhere around 20 to 30-fold weaker in potency. But we still thought this is something quite interesting to study because I mentioned to you that if you cut the whole C-terminal of the B chain off, the whole thing is negligible or the weaker in potency. But we probably can see that the Venom insulin is still quite a bit better than that. So that's why we decided to study that. So very early on, this is now 8 years ago now. So we basically, along with our structural biology collaborator, published this paper in a way that it suggests that there could be some mutations in the Venom insulin involved to basically compensate for the deletion of the C-terminal of the insulin B chain. And then using this concept, we make a bunch of different insulin kind of hybrids in a way, try to understand what are the potential roles of the Venom insulin play there. But for the interest of time, I'm just going to tell you that we end up developing an insulin, we call it mini insulin, in a way that basically the final A amino acid still cut off. So we should have been very low activity, but instead we put in 4 mutations we learned from the Venom insulin sequence labeled in red here, in a way that the whole thing can actually become something with a very similar potency as native human insulin while maintaining the monomeric state. This is a busy figure, but just to tell you that they actually act just like human native insulin in a way that all 4 mutations are required, because if we remove one of the 4 mutations, we will lose the potency by somewhere between 4 to 10-fold ish. And then in terms of the signaling, they actually look quite similar to native human insulin. We have a model try to explain that, because of course we don't have the C-terminal of the insulin B chain here, which is very important for the binding. So what we believe what happened is the 4 mutations are playing different roles here. So one of the tyrosine mutations partially compensate for the role of this deleted segment. And then in addition, there are 3 additional salt bridges potentially is involved to basically provide the same receptor binding activity here. And then so in addition to the original Venom insulin, our marine biologist collaborators, they actually find out even more sequences. And this time it's actually something quite different. Instead of having the same mutations on the AA, A9 area, they actually found that they can potentially have a longer extended insulin agent. So normally insulin ends in the 21st residue here, but as you can see that these are residues they can have, it can be quite a bit longer, with at least 4 to 5 extra amino acids. If some of you are familiar with the insulin-like growth factor sequences, you will know that IGFs, they also have, they call it extended D chain there. So it's a little bit similar to that, although in the IGF case, the D segment, they doesn't really get involved a lot with the receptor binding. So at that time, we thought it would be quite interesting to see what these residues are doing. Because this time we actually happen to know a bit more about the chemistry now, so we actually very fast we actually identify that there are some residues we learned from the Venom insulin. We can directly put it in, and then still with a shorter insulin B chain, we very soon were able to find a sequence. We call it HALQ or VH-ins HALQ because we create the venom insulin hybrid peptides. So again, busy figure here, but just to show you that basically the newer version of monomeric insulin HALQ, they actually bind to the insulin receptor with pretty much the same binding affinity like human native insulin. In terms of IGF-1 receptor binding, it also similar to human insulin, has a much weaker IGF-1 receptor binding affinity compared to native IGF-1. We further solved the structure this time using QuidelEM through our structural biology collaborators, trying to understand how these new version of monomeric insulin, although without the C-terminal of the B chain, how do they have the same binding affinity here? There are more about the structural biology, but let me just show you one of the key findings here. I mentioned to you that the B24 phenylalanine is very important because it's having a direct high hydrophobic interaction with the receptor. So what's really special about this now, the new Venom insulin, human hybrid insulin, is that the extended A chain, especially these leucine here in the A23 residue, is occupying exactly the same 3D space to basically totally compensate for the same hydrophobic effect here. So this is truly amazing in the way that basically these Venom insulin, they just evolve a new way, go through the extended A chain to basically totally mimic the same way that the million, if not hundreds of millions of evolution of the native, the mammalian insulin sequences, and then basically in the end can have the same biophysical property here, do the same interaction. And again, this is something we have already published here, but kind of just to show you about the monomeric feature here. So this is done by ultra analytical centrifugation. So the disulfide peptide insulin here, DOI, basically it's human insulin, but without the last A amino acids in the insulin B chain. You can see that it's a pretty good monomeric looking thing. When we do the human insulin in somewhere close to 1 milligram per ml concentration, so you can see that there's a mix of dimer, tetramer, and hexamer right here. So basically, this is kind of a pretty good control to see. We also do it with the insulin Lispro, so you can see that it's somewhere between hexamer and tetramer. So you see a good peak here, but there's still some monomeric and dimeric region here. In our Venom human hybrid insulin, we are seeing something that's pretty much purely monomeric here. And then just to further show you about this. So now the question is, can I talk about potentially monomeric insulin has the promise and has the potential to become something that's fast acting and potential shorter of duration action. Can we actually try to do that? The first thing we need to deal with is actually the stability. So there's a reason why native insulin want to be hexameric. This is because in the hexameric form, they can basically pack the hydrophobic region of the insulin together such that insulin won't be that aggregation point. So this is something to show you that the monomeric insulin we have, that's not just called HALQ because it's easier to call it. When you store it in a vial, and then with some gentle shaking, with some and then you measure the aggregation state. So basically, you can view that when you see a huge signal increase, this means that there is some significant aggregation there. So you can see that very fast, around 10 hours, things will get aggregated. In the same assay, if you do it with human native insulin without zinc, so not trying to push the hexameric state, we normally see somewhere between 20 to 30 hours. It was same assay, same condition. You will see it to become aggregated. So as you probably can see, this is something that become pretty easy to aggregate. And of course, this is not going to be helpful. So Yunshen Zhang, who is a postdoc in the lab, she actually developed some inulin base. So these are basically dietary, sugar molecule in a way that she developed some excipients based on they try to stabilize it. So as you probably can see that in this particular form of the excipient, she can stabilize it quite well. So we thought that, okay, now we at least can have something that's stable enough, will be good enough for us to evaluate what they are doing. We
Danny,
actually,
excuse me, so these excipients that she used were based on just that molecule to the left of her photo that are shown, or
yes,
did she
so they
throw
are
the
based
zinc
on
and others?
this. Yeah, so we actually do not add zinc, and to be honest, zinc is not it's not really an issue here. Let me tell you a little bit, because zinc requires this B10 histidine to coordinate in the way to form hexamers. We actually don't have this. So whether you add zinc or not, it actually doesn't make any difference at all. So zinc is not a thing. We actually add some, things like metacresol, things like that in a way that those are commonly used in insulin formulations to further stabilize the formulation in other commercially available insulin formulation as well. But basically, the main thing is this sort of inulin-based excipients.
Got it.
So we thought we have something now stable enough. We're just going to see whether they can do something. Fortunately, what we are seeing is now so this is just my theory because we just want to see whether they are still active. We actually see something that's actually more slow acting and more longer acting. So this is actually compared to the mother molecule. So it's actually the opposite of what we are looking for. And then we actually try to figure out why. And then by doing an MMR study, we actually found out that there's a direct physical interaction between the insulin analog and the excipient. So basically, instead of having insulin that's monomeric to get inside the body and potentially hoping them to be absorbed, now it's actually bringing the excipient there, so making it even bigger and more difficult to move. So we actually thought that we will need something else. But Yunshen was able to develop a newer version in a way that we don't see the direct interaction there, but still maintain the stability. So we now decided to give it a try, and then to really do a try to see how fast these things got absorbed, you really need to go for a pig model of diabetes. So in this particular case, these are Yorkshire pigs in the range of 50 kilogram-ish. And then this model at Stanford, it was originally developed by my colleague Eric Apple's group, and then we learned a lot from group members from his group and try to do it. So basically, we actually found that you will need to use a model like this to basically differentiate the different the fast acting properties of the insulin. So basically, in mouse and rats, we actually cannot even see a difference between Fias, Monomeric, and Humulin. But here, you probably can see that although we don't see a statistically significant difference between Fias and Novolox, but we are actually seeing a difference between the faster insulin versus Humulin part. So we decided to use this model to see whether we can see that, to have a direct comparison between Fias and our monomeric insulin formulation to see whether we can really see a difference here. So this is the final.
One quick question, just to serve before we advance, is from Ganesh Sable, what kind of interaction stabilizes the mentioned insulin with the shown excipient? That's like 2 slides back.
That's actually a great question. I will actually need to just go back, but this actually will be a good one to talk about it. So it will be quite similar to these figures shown by my colleagues Eric Appel's group. So what we believe is happening, we have some data to show that it's basically the excipients is basically having I would say more amphiphilic interaction with the insulin, such that it shielded the insulin from in a way that their hydrophobic parts will be stabilized by the excipients and prevent them from interacting with another hydrophobic region of the insulin to promote the aggregation. So basically, it's a way of you can have something to basically surround it surrounding the monomeric insulin in a way that they will not bump into another monomeric insulin to induce the aggregation. So this is what we believe what's happening.
Thank
all
you.
Right. Go all the way back. And this is the final slide the final data set I'm going to show you. So basically, as I say, we are comparing between Fiasp and our monomeric insulin formulation. And what we are seeing here is there's actually no significant difference between Fiasp and our monomeric insulin in the onset part. So whether it's the time to reach the nadir of the effect, or it's time to reach the 50% peak down. It's actually pretty similar. We don't see a difference there. So this suggesting that basically they are potentially equal fast acting in our current study, but we actually see a pretty significant difference in the duration of action, or basically you want to call it offset action here. So whether you calculate from from the nadir to 50% of the offset, or versus the full duration of action that's basically counting 50% down to 50% up, we are seeing a very significant reduction of duration of action here. And then basically what we believe this is related to basically the monomeric state in a way that to reduce the depot impact there. I also want to put a little bit comment on the similar duration of action part. So really, either for Fiasp or for Nunjaf, there are excipients they put in, try to basically, in a way that just to put it in general term, create a bigger pore between the blood vessel wall in a way that we have things to get infected, whether do vasodilator or do niacinamide. We don't have any of those things here in our monomeric insulin. So I actually, although there are different ways to explain that, I view this similar onset action result to be something pretty positive, because that actually means that if we use some of those excipients, that's already demonstrated by Nunjaf and Fiasp to have faster onset action, potentially add those things in our monomeric formulation, there is a chance or potential that we can see something even fast acting, although that will remain to be seen in future investigations. And finally, just want to talk about the summary of my talk today. So there's tremendous progress in the past 3 decades to develop new insulin analogs that's already providing tremendous clinical benefits for people with diabetes, both type 1 and type 2. And there remains a need to develop new insulin with both faster onset action and also shorter duration action. And basically, we developed monomeric insulin, which is inspired from the constant Venom-insulin to have a deleted C-terminal of the insulin B chain region, but still with the full potency. And then compared to hexameric insulins, the intrinsic instability of monomeric insulin require new excipient to stabilize it. And we have to put some efforts there. And finally, we were able to see that the monomeric insulin formulation, while demonstrating a similar onset action compared to Fiasp, but it's demonstrating a shorter of duration than Fiasp. And finally, just want to tell you that last year, actually, or a year and a half ago now, my colleague and I, we actually wrote this review, which is actually very interesting. We talk about, in our case, there's the Venom-insulin inspire human monomeric insulin, and a lot of you will know about the Gila monster story that lead to a lot of the GLP-1 and analog stories. And then we actually talk about there are a bunch of different, more unconventional molecules that lead to potential different hormones. We call it repurposed hormones. So if you are interested in this topic, feel free to read this review because this is something we thought that's quite interesting. And that's it. Thank you very much for your attention.
Thank you. This is fascinating, new /old inquiry. And I want to open it up to the audience to ask some questions. Please drop your questions in the chat, or you can unmute yourself or ask to unmute, and I'm happy to do that. Raise your hand. I had a couple of questions also, just kind of, prompted by some of the early comments in the chat. Can we talk a little bit about the insulin market and sort of the, pressures on getting new insulin to market? I mean, there was earlier, it was noted in the chat that, there's been a couple of insulins, Lilly's, Peg, Lispro, and Novo's NC0123-0327 were both live liver-preferential insulins that reached clinical trials but didn't reach market. Has there been drop off, in between sort of the generation of new molecules or new strategies and, getting them to market? And what is your thought on that?
This is a great question.
Anyone can weigh in. Actually.
I thought, potentially in terms of the marketing for some of the audience in the either Novo or Lilly who have strong ties with them can potentially provide some insights about this. These in terms of new insulin development, these require, I would say, way more resources, potentially to go do the regulatory process in a way that, there are more trials to go for compared to, say, things like Lyumjev or Fiasp. Those are newer excipients, but still the same active pharmaceutical ingredients. So the bar or the resource you put in to get them to be approved is quite different. Now you think about, potentially a fast-acting insulin with shorter duration of action. This is probably something that will be more suitable for, say, let's think about the market. I'll be more for type 1 diabetes market compared to type 2. And, we know that this is compared to type 2, a smaller market and potentially, scientific rationale is there to drive something like this, and whether the business rationale is still there for the farmers to consider to push it. That's probably another consideration for them. If I want to say what would be something that it's pretty obvious that everyone is interested, that will be, I would say, glucose sensitive insulin, because that's something that potentially can work for all the people with diabetes, and including type 1 and type 2. So of course that will be something that everyone will have a strong interest there. Another thing I am interested in will be more in the oral insulin, or something that's a insulin-like molecule that can be administered orally, because this is also going to be something that will attract a lot of interest from both scientific and potentially business perspective.
Easier compliance.
So I will say that, really from my angle as a scientist, everything when there's a need, I think we should work on that. And but there's another spectrum for people to think about whether there's enough rationale or resource or motivation for them to do it. And I'll say that's probably beyond the scope of what I can think about for my current position. And you will be good to see if the audience have some other thoughts to share related to
Yeah.
this.
We do have an input from Thomas Hoag Jensen. Although a BIL gave lower risk of hypo and slight weight benefit, there were long-term side effects on the liver, increased liver enzymes, triglycerides, and signs of fatty liver. NN327 didn't go far, only phase 1 as he recalls. So that's kind of interesting. I wondered, looking at all these sort of like potential model systems up here, do snails or any other of these other animals exhibit autoantibody situations versus their insulin, or if specifically the insulin beta chain, the B923. Are
That's
these
a terrific question.
is there any circumstance where you could manufacture that?
That's a terrific question. So we never really test the venom in the native venom insulin by itself because the sequence homology is low and it's really not that active as well. But we actually did test the antibody response of the mini insulin. So basically the human version, the human monomeric insulin with the 4 mutations there. They do have actually 2 of the mutations from the B9 to 23 range because there's a B10 and B20 mutation there. In that case, there's a 21-day immunization. We don't see significant autoantibody generation for that molecule. And that's what we have been doing so far. And this is a fascinating question because in the end of the day, normally if you really want to know whether there's really antibody, probably a phase 1 trial in human will be something you will know the most. And then this is also really, really difficult to predict. One of the relevant, not in the insulin area, but something relevant to the field is Zealand has this soluble glucagon analog that basically out of the 29 amino acid, they have 7 mutations. So, from if there's a scientific group try to propose something like that, and you want to put it into NIH review, chances are you will get comment that this is pretty much guaranteed to have immune response, and then probably you will get killed. But they do all the trials. It's now FDA approved, and they doesn't seem to have very significant immune response. Although 7 out of 29 amino acid got mutated in a small enough single chain helical peptide, so that's still okay. So this is something pretty difficult to predict, right? So you probably will need to do a trial to understand that.
Yeah, just sort of throwing out big ideas. And then I guess sort of this is sort of a bigger question too. Have you or the field thought about creating an insulin that provides physiological mechanism, but it's not seen by the immune system, right? So are you thinking about when you're sort of changing up a little bit of some of the amino acids here and there, adding some of these excipients and so forth, how might you cloak it from the immune system for those that already have the insulin autoantibodies?
This is a terrific question. I really like this question. There's pretty well known insulin like, well, not insulin like, but basically a non-insulin peptide activator of the insulin receptor. It's called S597. It was originally published by Novo Nordisk, and it's basically a single chain peptide sequence in a way that they, just like insulin, they bind to the 2 sides of the insulin receptor in a way to activate it. And for some reason that peptide never got developed further into clinical studies. So, other than their behavior in cells and in animals, I don't really know what they can potentially achieve in human. But because of that molecule as an example, I think it's actually possible, and potentially this will be an area of interest for us as well to develop a non-insulin molecule that basically will fit in what you just said. You will
Yeah.
basically also bind to the receptor, reduce the blood glucose levels, but it's just not insulin at all. So such that you can avoid a potential anti-insulin antibody problem.
Hmm. Even if, in context of the islet implantation, if you could have some of those, re-engineered beta cells cranking out a cloaked insulin, might be helpful.
That would be
Um,
something pretty cool to see.
yeah, very cool. And then just the last question I had for you, there's this whole cohort of individuals who ascribe to the high protein, low carb diet. They take our insulin, they follow sort of this Dr. Bernstein. And it's interesting because our insulin is very slow acting. And, can you imagine sort of like a variety of insulin that would be diet sensitive in the future? So for those who are, deciding they're going to, just be protein based or non-protein based, would there be advantages or disadvantages to having insulin, particularly for the T1D crowd?
Okay, that is a interesting question. I actually never thought about this, whether there could be different insulin that can address different diets.
Hmm.
Well, it's pretty difficult for me to imagine how to implement that right now. I'll put more thoughts about it, but that's an interesting thing. I would say
Yeah,
in
I
the
mean,
ideal
they only
scenario,
use the R insulin, which is the old, the old slow version, because they match it to the digestion of the protein. And they have the ones who really abide by it have some pretty good results.
yeah, in an ideal scenario, kind of let's think about something kind of more long term. Remember that there's actually let me go back to that one. I actually really like that challenge theme. So assuming we solve some of the problems here, you got all these the current ones to be like the past ones in a way that you address all of this. Say if you really have something that's really faster, like pretty fast acting insulin, we'll say you have a injectable insulin that has the same kinetics of the inhaled insulin property. Potentially, you may argue that the AP, the closed loop system may be able to be something that you actually don't worry about what you are eating, but basically, even if you are eating something that's taking a longer time to be absorbed, but you just see whatever the trend of the glucose levels from your CGM, and then there's always a on-demand fast on, fast off insulin to address that. It could be due subcutaneous, or some of you may know my colleague Rehan Lau. He's very interested in those implantable pumps, such that insulin can be delivered IP in a way that could be something even faster, and then potentially shorter duration as well. So one way or the other, I think if we solve some of the current challenges, we should be able to imagine something that's much easier and tighter control of the glucose levels.
Good, great answer. Here's a question. Two questions, actually. One from Ganesh Sable. Based on your SAR studies, any thoughts on critical insulin mutations, especially the last few residues of the C term of B chain towards biasing IGFR receptor versus IR affinity?
Yes, this is a terrific question. We actually, so this is a terrific question. So of course, whatever insulin analog you go for, you worry about IGF-1 receptor binding. And then this is actually a good thing to show it to you. When you cut the last 8 amino acid off, in the C terminal of the B chain, you can actually, so this peptide insulin will actually bind even further away compared to native insulin to IGF-1, because those are important residue for insulin receptor binding. They are also important residue for IGF-1 receptor binding. So basically, when you cut that off, you are almost certain making the whole thing better. So probably that's why you can see that even when we put in some mutations that are known to engage with IGF-1 receptor, but we still overall, at least for this molecule, it still have an even worse binding affinity to IGF-1 receptor. So theoretically, if people want to avoid the binding with the IGF-1 receptor, cutting the last A amino acid off from the insulin is a good approach. But the thing is, in that case, you will also get rid of quite some of the insulin receptor binding. So you will need to have some mutations to contribute to the other part in a way that you can still see similar binding affinity.
Great answer. I would just add this commentary that's in the chat so that those who are listening later can hear it. Yeah, from Dr. Hoag Jensen, the idea of finding an insulin mimetic was mainly aimed at oral delivery, but since S597, et cetera, are almost as big as insulin, there was little hope of better oral bioavailability. S597 may help autoantibodies, but isn't that a rare problem? S597 may also raise antibody itself being non-human sequence. Do you want to comment on that?
Yeah, I think that's totally spot on. In the way that it probably pretty rare in a way that the autoinsulin antibody in a given patient it's such a huge problem in a way that they cannot even take human insulin or all those analogs to address their blood sugar need. So that's probably the difficult part. That's probably the rare part. And then in the case of S597, I mean, Thomas used to work in Novo, so that's probably one of the reasons behind why Novo never really push S597 further in the clinical studies.
Okay. Well, thank you so much. I really, we went a little over, and I really appreciate you giving us the extra time. This was a phenomenal talk and really exciting times for, this kind of approach. And it'd be very interesting to see what comes next from your lab. So appreciate you joining us today, Danny.
Thank you, everyone.
Have a great rest of your day.
Thank you. Bye-bye.